Hi, my name is Megan Sage. I'm a third-year PhD student in the lab of Dr. Diane Duffy, and I'll be presenting my research on how neurotensin stimulates theca steroidogenesis in the monkey ovarian follicle. One of the central components of the study is neurotensin, which is a tridecapeptide found to be produced in the ovarian follicle during ovulation and is essential for ovulation to occur. Ovulation is the process by which an oocyte is released from the ovulatory follicle. As you can see in the image set on the right, abrogation of neurotensin prevents rupture of the follicle and leads to the formation of a hemorrhagic cyst. Apart from the oocyte, ovarian follicles are comprised of granulosa cells and theca cells separated by a basement membrane. Granulosa cells produce neurotensin in response to the ovulatory surge of luteinizing hormone, or LH, and they express neurotensin receptors. Theca cells surround the follicle and are responsible for producing androgens that are converted by neighboring granulosa cells into estrogens. Neurotensin's essential role in ovulation is important due to its potential translatability to birth control and fertility research. However, the mechanism of action of neurotensin within the follicle during ovulation has yet to be uncovered. Due to the importance of paracrine signaling in the follicle, as well as the dependence of the ovulatory sequence on local and systemic hormone levels, we turn to follicular steroidogenesis to investigate the function of neurotensin. Steroid production in the follicle is collaborative in that theca-produced androgens are converted into estrogens by granulosa cells. Theca steroidogenesis is known to be stimulated by LH or human chorionic gonadotropin, or HCG, via LH receptor or LH-CGR signaling, as well as by forskolin via receptor-independent activation of the protein kinase A or PKA pathway. Our twofold hypothesis in this project is that 1. Neurotensin is a novel regulator of ovarian steroidogenesis, and 2. Neurotensin acts on theca cells to stimulate androgen production. In our investigation, we utilized an ex vivo theca tissue culture system in order to study the effect of neurotensin on theca steroidogenesis, with HCG and forskolin serving as positive controls. Ovaries were removed from adult female cynomolgus macaques after a modified ovarian stimulation protocol, and the theca layers were isolated from large antral follicles and minced to uniform size. RNA was isolated from cultured theca tissue and analyzed for expression of mRNA characteristic of theca cells, specifically STAR, CYP17A1, and VEGF-A, after reverse transcription and qPCR. Expression of all three transcripts was confirmed, shown in the bottom left graph. Theca tissue was treated for 24 hours, and media was then analyzed for androstenedione, or A4, and progesterone, or P4, concentrations via ELISA, or the ADVIA Centaur immunoassay system, which were normalized to the DNA content of theca tissue. These are the data showing theca tissue androstenedione production in the top panels and progesterone in the bottom panels, with the y-axis showing nanogram hormone per microgram DNA extracted from tissue, and the treatment group in the x-axis. Starting on the left, both HCG and forskolin treatment increased androstenedione and progesterone in media of cultured theca compared with untreated cultures, which were the expected results of our positive controls. In the graph in the upper right, our novel findings show that neurotensin increased androstenedione concentration in a dose-dependent manner. In contrast, as shown in the lower right, media progesterone concentration was unchanged in response to neurotensin at any dose. Together, these results indicate that neurotensin stimulates steroidogenesis in theca cells. While HCG and forskolin increased both androstenedione and progesterone, neurotensin treatment showed a selective increase of androstenedione only. Given that both HCG-stimulated LH-CGR signaling and forskolin promote PKA-mediated stimulation of steroidogenic enzymes, the contrasting androstenedione selectivity of neurotensin may be due to the use of a non-PKA mechanism of steroidogenic regulation. Looking at the process of steroidogenesis in theca cells shown in the graphic, the production of each steroid is dependent on its direct enzyme as well as the production of steroids upstream in the pathway. Neurotensin increasing androstenedione and not progesterone could therefore be due to a selective augmentation of an androstenedione-specific enzyme such as CYP17A1, which we've confirmed the mRNA expression of in the theca tissue cultures. Future studies will continue to investigate the role of neurotensin in follicular steroidogenesis through its action on theca neurotensin receptors and its effect on steroidogenic machinery and cholesterol utilization. Thank you.